My name is Alan Van Dynes. I'm with the University of California, Davis, and I'm part of the SOLCAP project, which is sponsored also by the USDA. So basically, this whole workshop is sponsored by the USDA. Okay, so I have the first talk, and we have a series about 6 talks today, and we're going to talk about try to use the data that we're currently generating in SOLCAP, and also with the tomato draft genome sequence. So how can we best use that as plant breeders and take advantage of the data and give you a quick update of where we are with that data. So I have the first talk, and I'm going to talk about really sequencing technologies, and part of it is for, as breeders, you don't have to think about sequencing technologies so much, but I think it's important that you understand the differences between them, because that really qualifies the type of data that comes out. And really that's what I want to try to relay today, because not all sequences are the same, as you know. So how to sequence, or what are the new technologies that are coming out? What part of the DNA you sequence is actually important. You want to sequence genes, or want to sequence random parts of the genome. What line should I be sequencing? And that has actually it's pretty relevant as far as how useful the information coming out will be to your programs. And then how many lines? And so I used to say, the argument used to be, what line should I sequence? And a big argument, and everybody had to come up with a consensus. But with technology today, it's how many lines and which lines or which populations do I need to sequence. And so that really changes how you can think about things. So with sequencing, unfortunately, we don't have a technology right now that can basically take a DNA molecule and sequence it from one end to the other. So we have to do, basically what we end up doing is we chop up the DNA and we try to put it back together. And that has some issues, and different technologies handle that in different ways. So I'll call it the first generation technology, which is a Sanger or dideoxy sequencing, that anything from 5 years ago and before has been done for the most part, including a big part of the tomato genome as used. Then there's second generation, or they call it next generation sequencing, and I'm going to talk today about what we're calling third generation sequencing now. So Sanger sequencing, basically, the DNA is actually cut with restriction enzymes. It's actually cloned into a vector, and with primers that are specific to that vector, we can start a sequencing reaction. And the dideoxy method, which is it's an NTP, so an ATP, A, G, C, or T that actually has an end on it that stops the sequencing reaction, is dideoxy. And where it ends is random. And because it ends at different places, you can get basically a sequence when you run that through a capillary gel. And so and you get about 650 to 1,000 base pairs in a given run or read. So that's how the human genome was done. That's how rice was done, Arabidopsis, kind of the first things that were sequenced. Tomato was actually done with a combination of all these technologies I'm going to talk about. So something that's changed, so this was the first one I talked about, is quite expensive and labor intensive and takes quite a while. Something that's changed, the next technology we came along is called 454 pyrosequencing, and this takes advantage of basically the luciferase gene, which is the gene that makes a firefly glow. And they use that as the detection method in an enzymatic reaction. So instead of cutting with a restriction enzyme, they actually shear the DNA into single strands. And they'll put on adapters on the end of those DNAs, and those adapters are the primers that drive your sequencing reaction. The difference between this step and the last one, and really all the next sequencing technology we'll talk about, is there's no cloning step. And that changes, makes things a lot easier, and allows you to basically sequence things in a highly parallel manner, as opposed to one clone at a time. The sequence is amplified on these little beads, and you get you call emulsion PCR. And by adding single nucleotides one at a time and looking at the light reaction coming off from adding a specific base at a spot, you can get a sequence from that. So this is highly parallel. You get thousands of sequences simultaneously, about hundreds of thousands. Another technology came out about the same time. It's a company called Solexa, was bought out by Illumina. Slightly different technology called sequencing by synthesis. And what they do is they start with the same method, essentially. They shear the DNA into pieces and add some adapters. They go through a PCR amplification, and you get these loops, which then are linearized. And so these little blue and purple bits on here are really adapters or primers that the sequencing reaction starts on. And instead of looking at things one base at a time, they add all the bases at once, but each base has a different fluor on it, so a different fluorescent tag on it. So based on the color of that tag, you can tell what base was added at each cycle. The more cycles you do, the longer your sequence. Now within a limitation, and I'll show you those limitations later. And so Illumina sequencing, that's based, the last two are called next generation sequencing, and the next 2 or 3 that I'm going to talk about are stuff that really is just coming out. It's in beta phase, but it can change a lot of things that we're going to do. There's a company called Helicos. They call it true single molecule sequencing. The difference between it and Illumina is that it's very similar to Illumina, that they basically shear the DNA, have adapters, have fluorescently labeled nucleotides that they add, but there's no